Well, thank you very much for the nice welcome. I'd like to thank UMass Lowell for having me, and to the Mass Bioed Foundation. They do a lot of great work around the state. And working for Millennium Pharmaceuticals, I actually participate in 3 different volunteer events that they do, working with students of all ages, which I'll get to a little bit before. And I want to thank you guys. And I want this to be an interactive discussion, so please, if you have a question, raise your hand, just let me know. It'll probably be easiest to take them all at the end, but if you have stuff, please write it down. I'm not leaving the stage without at least one question. So I wanted to ask you guys, first to start off, has anyone ever met a scientist here before today? Couple? All right, most of this side, yes. All right, well, after today, everybody's met a scientist. Everyone in the back of the room, myself. So I want to give you a little bit of a background. So I work at Millennium, the Takeda Oncology Company. We're a Cambridge, Massachusetts-based company. We're Millennium Pharmaceuticals, and then about 2 years ago, got bought out by a large Japanese biotech company. And I'm a scientist, research investigator, and I want to talk to you today about cancer drug development. So we're a cancer research company. So let me tell you a little bit about, first, what I do. Hopefully this will keep working to kind of have a little bit of a delay. Bear with me. Might have to do a little song and dance in the meantime. I don't think anyone would appreciate that. Okay, so I want to tell you a little bit more about who I am personally, kind of give you an idea. You might have some ideas already of what a scientist is like. Hopefully I'm going to end up crushing some of those myths. So I also want to tell you professionally about what I do in the lab and at my computer, and how exciting a career in cancer research can really be, how rewarding it is too. So I want to talk about that. You've probably studied cell biology in class. I don't know if you've ever studied cancer biology. Have any of you looked at cancer cells at all? Any cancer biology background? A little bit? I want to give you a quick background and then tell you about the drug development process. So this is a long process, involves testing cells, a bunch of different measures before you go into testing new medicines in human beings. So I'm going to give you a background on the entire process. New term called personalized medicine. So this is a new term within the industry, and it's a very exciting area of research. It involves giving the right medications to the right people. So if we give a new medicine to everyone in this room, and let's say only the women respond well, all the women, the disease is cured, because guys, we know they're better than us anyway. So all the girls are cured, but we're not. So what I do at Millennium is study the genetics behind it. Why are the girls responding well to the medicine? Why are the guys not? I want to tell you a little bit about this new compound. It's a new medicine by a company called Lexicon. So the medicine is called PLX 4032. The reason why it has a number is because it's not on the market yet, but it's showing really good results within patients right now. And then I want to talk to you a little bit about why I chose a career in drug development, specifically in cancer development. It's very, very personal to me. And as Suzanne mentioned, I'm very, very passionate about it. And I know everyone in this room has some sort of tie to the cancer diseases. Whether or not you know someone who has it right now, who has passed away from it, it's very, very personal. And it is for me. That's why I got into it. And that's why I stay in it and work hard every day. So definitely want Q&A at the end. A little bit about me. Bear with me. Well, I know what's coming up. I was born in the city that never sleeps. I'm a New Yorker. I grew up there for most of my life. And this is still not coming up, but bear with me. As a child, I was always very close with my parents. We always had a very special relationship. My mother is a teacher, and my dad is a lawyer. So how did I get into science? Well, they kept pushing science on me big time. Growing up, they always kind of had a passion for it, and teaching as well. And one of my earliest memories being involved in science was from my father. As a lawyer in New York City, you don't expect him to be involved in science. But in 3rd grade, when we had to write a school paper, in 3rd grade, he kind of pushed me to do one on DNA. What kind of 3rd grade writes a paper on DNA? I didn't know anything about it. Had to research a ton, but because of his pushing, here I am with a career looking at DNA every single day. So he had a huge impact on my career. There we go, city that never sleeps, New York. So that's where I grew up. After growing up there all my life, I decided to stay in New York, but I went up north to college. I went to the University of Rochester, where I didn't know what I wanted to study. I knew I had a natural interest in science. I always was interested in it, but I wasn't quite sure how I could apply it. You know, you guys have a unique opportunity in high school. I didn't know. When I thought if I studied science in high school and in college, all right, well, maybe I'll go pursue a career in medicine. Maybe I'll go to med school. That ended up not being the right route for me. So at U of R, I really was able to explore all these different avenues. I knew I had a definite interest in science, so I studied science, but I declared my major at the end of sophomore year. So you have plenty of time. What you really want to do is try to figure out all your different interests. Again on the delay, sorry about that. So I ended up declaring a major in cell and developmental biology, and that's molecular cell, genetics. It really encompasses a whole lot of different areas, but plays a big role in what I do today. But as the previous panelists described, you know, college is your time to explore every single thing that interests you. I wasn't just a science person. I was actually a journalism minor. I loved different extracurricular activities. I actually had a radio show on campus. I love music. I'm very passionate about it. So I had a ball doing that, communicating, you know, getting on the air and just being able to communicate to the entire city. It was a pretty powerful and cool way to get some ideas across. So I would just invite my friends into the studio, and it was a really good time. Very into sports. So in college, I was able to pursue that. One of my biggest interests is weightlifting. Aside from my full-time job as a scientist, I actually work as a part-time personal trainer. So you're able to explore all different areas that are interesting to you. Sorry, my little animations aren't coming up. After college, I ended up moving to Boston. There were a lot of different reasons why. I wanted to stay on the East Coast, but wanted to try out a new city. I've been in New York all my life. Don't get me wrong, I love it, but I love Boston too. I've had a great time here. There's the DNA.
Here's the radio.
Perfect. Radio, music, sports.
Weights.
Boston. There we go. Thank you.
You want me to help you out here?
No, I'll advance it if I need. Thanks. So coming to Boston was a great experience for me. And I got a job immediately right after school at Millennium Pharmaceuticals, which was very fortunate. I mean, when I actually moved to Boston without a job and applied everywhere, sent my resume out to tons of different places. And Millennium, as I mentioned before, we're the Takeda Oncology company. Oncology is cancer research, and Takeda is a Japanese biotech that bought us out 2 years ago. I went back at night, in addition to my work, you know, as a scientist at Millennium, and got my master's in biotechnology at Harvard's Night School. That's the Harvard Extension School. So it's been a great experience in Boston so far, and I've been very fortunate. But as far as what I do right now, let's talk it through. So I told you I work as a cancer scientist. It's in the molecular medicine group, which is part of clinical. So clinical meaning patients. When new medicines go into patients, my job is to look at patients' different genetic profiles and try to understand who's responding to the medicines and who's not responding. So everyone in this room, we're all 99% the same genetically. So that 1% within the genetic code, those A's, T's, G's, and C's that we study in biology class, that's what I look at to try to understand the differences. If we can understand why, let's say, this half of the room is responding well to a drug and this half isn't, well, we want to give everyone who has their genetic profile the proper medicine. There I am. So that's me in the lab. In addition to my roles as a scientist, as Suzanne was so gracious to point out, I volunteer a lot of my time to educate and to talk to students. It's the fun part. Science is hard. You know, studying it in school, it's not easy. But my goal within cancer research is to help out as many people as I possibly can because I'm so passionate about finding a cure to this disease. Millennium's motto is "We aspire to cure cancer." It's a bold, energetic, in-your-face motto that's really putting it all out there. Finding a cure to cancer is not easy. No one's done it so far. We have different therapies to treat different cancers, but It's on all of our ID cards. We're constantly reminded of it. So I volunteer a lot of my time working with students, high school age in those pictures, and younger students as well. I went into a few classrooms this year to speak with some 6th grade students and even younger, as you can see from some of those pictures. So it's a lot of fun. I get a lot of, it's very rewarding with that. But I want to give you guys a background on cancer. You said you haven't seen a lot of it so far. So what exactly is cancer? I'll just tell you without my slides. So cancer is a big set of diseases that's characterized by uncontrollable cell growth. So you've heard of cancer, tumors. What all tumor is cells growing out of control, and they bunch up on the cell themselves. And it's because the growth signals are completely out of whack. You have different receptors, different proteins within the cell that can't stop growing. And what causes cancer is a little bit different. The simple answer is DNA damage. So again, I told you I study DNA. I study different mutations, deletions, chromosomal aberrations, all that kind of stuff. But it's caused by environmental factors and genetic factors. So the genetic factors, if your family has a history of having cancer, you are more likely to get it. I mean, I personally have that in my family. But it's a lot of other environmental factors that could make you more or less likely to develop the disease. So the ones you've heard, smoking obviously, ultraviolet sunlight, got to put on the sunblock, things of that nature. So we're going to go over a bunch of these different factors. So here's a picture of a normal cell versus a cancer cell. In a normal cell, you've got your cell membrane, nucleus, everything is intact, and actually a very precise shape and structure to it. You guys have seen this in class plenty of times. As I mentioned with cancer cells, the receptors, some are expressed more than others, some are downregulated or not as highly expressed. All of the growth signals are out of whack, which will lead to the uncontrollable cell growth. With normal cells, it's one nucleus per cell. And again, a precise structure, the cells are sticking together. Whereas with cancer, the cells are completely out of whack. You could have multiple nuclei per cell, and oftentimes the cells break apart from one another. We went over most of this already. So normal cells, when to reproduce, know when to stop reproducing, and they stick together, whereas cancer cells do not. Give this time to load back up. The tricky part with cancer cells is when the cells aren't sticking together, you can have what's known as metastasis. So you've heard of some people having a tumor in one part of the body, let's say the lung. Well, if one of those cancer cells, or many of the cancer cells, break out and get into the bloodstream, the cells can attach anywhere in the body and start growing again. So you can find lung cells in the brain, and lung metastasized to the brain. It's kind of, each one of these different cells has a distinct morphology, so you can notice when cells shouldn't be in the organ that they started off in. Now, the human body has, you have an immune system. You're taught to fight off diseases and colds. So with normal cell division, if you have a damaged cell, it'll undergo apoptosis, programmed cell death. It'll kill itself off. With cancer cell division, there's so many different growth signals that are out of whack, the body can't do this anymore, and a tumor piles up. So we talked about metastasis, which is my next couple of slides, which is when cancer cells break off and can attach anywhere in the body. But these are some images of cancer cells in culture. So this is what I look at on almost a daily basis, and they're pretty gruesome looking. These aren't normal cells. So when I show you the cartoon images, it's one thing. But when you actually look at cancer cells under a microscope, they have this distinct and really weird morphology where the cell membranes aren't intact. They get these little spindle, kind of spiky edges and ridges to them. And you can just tell that they look diseased and unhealthy. We talked about metastasis, so I'm going to go through some of this. As far as what causes cancer, again, DNA damage coming from family history or environmental factors. So family history, has anyone ever heard of BRCA genes? A couple of you? So these are two different genes that are associated with breast cancer. So if an individual has a certain variant or a mutation within the BRCA1 or the BRCA2 genes, it makes them more likely to develop breast cancer. It doesn't mean they're going to get it, but it poses an interesting decision, and it's a tough knowledge to have, knowing that you are more likely to develop disease later in life. There are a ton of environmental factors that cause cancer. Cigarettes make you more likely to get cancer. I know you've heard non-smoking ads all your life, but I've had to try to convince a bunch of my friends not to quit smoking. I mentioned that I'm very close with my mother. One of my best friends growing up, I had to watch his mother pass away at about 52 years old from lung cancer that spread to her brain, exactly what I just told you about. So it's a very serious thing, and it's very, very striking. Also, high-fat diets. We talked about UV sunlight. Sexual history and infection. There are different major risk factors for cervical cancer. So there's a bunch of different environmental factors that play a role. So if we know that genetics and environment play a role in developing cancer, what do I do as a scientist? What do we study every day? How is the science that you study now applicable to life and to getting a job when you get out of college? So we study all the different proteins and pathways that are involved in cancer, cell growth and controlling cell growth. The only problem is there are many different types of cancers, and each cancer is genetically different from each other. So not only are there different cancers of organs, lung, breast, brain, colon, everything, but if you take 5 different lung cancer patients, they have the potential to be genetically distinct from one another as well. So you can't treat one lung tumor the same that you would treat another one. It's very tricky. So we study the different genes and pathways that are involved in cell growth. So I want you to keep that image in mind because we're going to look at it a few times. And when we talk about cancer genes, I want you to think about a car and the accelerator and the brake. So there are 2 different types of genes that we think about with studying cancer. Those called oncogenes. So oncogenes promote cell growth. They tell the cell when to grow and how much to grow. Now, in cancer, imagine you're in the car. So that's the accelerator. If you're in the car and you just floor it and you've got glue underneath the accelerator, the car goes out of control. So that's what oncogenes are in relation to cancer. These genes are out of control, promoting way too much cell growth. Tumor suppressor genes are those that are supposed to keep the cell growth in check. So that's the brake on the car. So in terms with tumor suppressor genes in cancer, they've lost the ability. It's like pressing on the brake and nothing is happening. You can't control that cell growth. So as far as we study the different genetic pathways and the different protein pathways within the cell, but how do we get from looking at cells under a microscope to developing a medicine that will actually help patients? So if we go back and think about that picture of the cell that I showed you, where we've got hundreds, if not thousands, of different ways that cells grow. What we look to do is analyze those, and if we understand that, let's say there's this one protein that's overexpressed in cancer cells. If we're able to develop a medicine that can somehow block that protein, well, then maybe we'll see a decrease in tumor growth. Maybe we'll see the tumor shrink. Maybe we'll see patients feeling better and surviving longer. So that's what we try to do by looking at the different oncogenic and tumor suppressor pathways and looking at these different proteins and genes that are involved and viewing those as targets so we can develop medicines against those targets. So what I want to give you an example on with the drug development pathway, let's take the RAF protein, RAF. I'm going to show you a picture of it in the cell in a couple seconds. And well, first it's really important to realize how many proteins are out there. So this is a picture of the cell. You have different proteins on the cell membrane surface. You have different ligands that bind to those proteins. And once binding to those proteins, it sets off a cascade within the cell. All these different protein interactions that promote cell growth. And that send what are known as transcription factors to the nucleus. Basically, just a ton of different factors that influence cell growth. So how do we decide what to study? There's one of these pathways might be indicated, might be upregulated in one type of cancer, whereas in another cancer, it might not. So I want to focus on this protein RAF and tell you about an exciting new medicine that blocks a certain type of this protein. So again, we have our protein crosstalk. Bear with the computer 1 sec, it's a big file. And what this medicine is designed to do, so RAF is an interesting one. I'm going to tell you a little bit more about it after we go over the specifics of the cancer drug development cycle. But it's a protein that's mutated in about 40 to 60% of skin cancers, melanomas. So if we know that this protein is mutated in that many patients, well, if we develop a medicine that can block the mutant form of the protein, maybe that'll have some efficacy. So the drug development cycle, if we have a new medicine to block this RAF protein, well, what kind of steps and how much is it going to cost to get it to market? Because I'm sure all of you have heard before, the pharmaceutical industry in general costs a lot of money to do things. It takes a long, long time to get a drug on the market. Well, the first stage is the drug discovery stage. And it involves, once you have a target, so we've got our mutant RAF protein. Well, how are we going to develop a medicine to block it? We've already done that, and we want to make sure that it actually blocks the protein. So it involves a complex set of experiments to design this and execute the experiments. Once we know that our medicine actually blocks this protein by different experiments that I'm showing here, we have what's known as a DC, a drug candidate. That's what's called the drug discovery phase. Once we have a DC, we have either a pill or a capsule or an IV injection that's supposed to block this protein and decrease tumor growth. We first test it out on cells. So we'll look at cancer cells in a dish, what's known as in vitro testing, which was talked about before, and see if our molecule works to kill cancer cells. So the unfortunate reality of the biotech industry, as most of you probably know, is there is animal testing. And the reason for this is it's a very controversial area, and I like to speak to students about it to really be upfront so they understand what goes into the testing. And the reason why there is animal testing, as unfortunate as it is to try to mimic or mock a human system without going into humans just yet. If we test a new medicine on this entire room and everyone dies, well, that's a big problem. So let's say before going into humans, as a biotech company, we have to go test cancer cells in culture, so in a dish, and then in animal models, and that's known as the non-clinical development. Once the ethical tests that are done proceed, then we go into humans. As soon as we know that the drug is safe and that we know the appropriate dose. Yes, question, please. Great question. Great question. And you're absolutely right. Humans are more complex. And as far as non-clinical development, there are differences, but there's also a lot of homology between the two systems. So we basically try to understand the different proteins that are the same in mice, in rats, monkeys, different animals, to try to scale up as close as we can to a human system. But you're absolutely right. Before we get into actually testing in patients, there's still some unknown. And that's what leads us to clinical development. So there are 3 different phases of clinical development. Phase 1, we're basically just testing in a handful of patients. So we're not testing in a couple hundred just yet. And with phase 1 patients, in a cancer trial, these are normally patients that aren't doing so well. They've tried a bunch of different therapies, and unfortunately, their cancer has progressed. So we're trying this as kind of almost like a last-ditch attempt to help them out and benefit the patient. So it's in a handful of patients. It's designed to understand the toxicology, so the different effects that the medicine has on the body, and also to look at the dose that we established before and see if that dose applies in humans. So we're looking for the maximal dose that we can give a patient and to help kill the cancer cells. Once it passes, if our medicine is doing really well, we test it out in more patients. And the phase 2 trial, it's really designed to understand, so if we're blocking, remember our RAF protein, that's the example, well, is it doing it in patients? So what my group actually helps to design is the different experiments that, okay, well, if we think that we're blocking this protein in patients, if we take either a slice of their tumor, so a tumor biopsy, or even a blood sample, extract the DNA from that and look at it, well, is it actually doing what we think the drug is supposed to do? So that's in a lot more patients. Now, phase 3, this is where we really know a whole lot more about our drug. And if we have this new medicine going into phase 3 trials, we want to compare it against the standard of care. So RAF, we were talking about melanomas. Well, we want to go right up against what patients are currently receiving and say, is our new medicine better than the standard of care? So this is all clinical development. Once clinical development is done, if our medicine still shows a lot of promise to benefit patients, we submit an application to the FDA with all the safety data, all the experiments, and data surrounding the experiments. Submit an application and hopefully receive approval. Now, Millennium, I showed you one of our major drugs over here. It's called Velcade, and it's designed to block a certain cellular protein called the proteasome. So the proteasome is kind of like the recycling bin in the cell, and it chews up old proteins and spits them back out for the cells to use. What our medicine does, it blocks that protein, which causes the cancer cells to be confused and die. So this medicine right now we have on the market, and it's used to treat two different types of blood cancers, multiple myeloma and mantle cell lymphoma. Millennium's a company of about 1,200 people. We're in Cambridge, Massachusetts, and we're a mid-sized company. We're not even a big one. We're not even a large pharma company. This entire process, because we have to go through so many hurdles in terms of making sure the drug is safe, making sure we know exactly what the drug does, it takes a long time. It takes a lot of money. When we're testing out, designing these experiments, it's a very, very long process. It can take over 10 years, cost upwards of $1 billion. Now, if your drug is showing amazing results right off the bat, there are ways to get it into the clinic sooner. So Velcade was showing such good results in patients, it was given fast track approval. So it took about 4 years from the first clinical study. Again, this is just showing the attrition in drug development, where you can start off with so many different ideas of molecules. Well, if we block this protein, block that protein in cancer cells, that it'll benefit patients. But if the drug isn't safe, if patients aren't showing good results right off the bat, we have to cut the program. If it's not showing good results on cancer cells in a dish or in the animal models, the program is done. So there's a very strict criteria. And as you can see with the clinical trials, we tested more and more patients before it can actually get approved. So I want to speed through this a little bit because I know we're running low on time. So we talked about personalized medicine and the fact that this RAF protein is mutated in about 50% of melanomas, skin cancer. Now, when a patient is diagnosed with malignant melanoma, this is one of the worst diagnoses that you can get. And the prognosis isn't good. The average survival time, I believe, is about a year or two. So this company, Lexicon, came out with a novel therapy, a targeted therapy. In the past, drug companies, if you go into a doctor and they diagnose you with cancer, the treatment is more of a trial and error basis between chemotherapy, radiation, different types of therapies that are basically a trial and error basis. If the patient does well, great. If not, if all these devastating side effects, it's really hard to understand that. And it's hard to watch a patient go through that. So what personalized medicine is designed to do, if we can understand the proteins and the genes that are involved in cancer, like this RAF protein, we can develop medicines to it based on a patient's genomics and proteomic profile. So these are a list of drugs that are on the market right now that target specific proteins. They're not just going in there to try to kill all cells, because when you kill cells, you're going to get some normal ones too. And that's where all those vicious side effects come from. So these different drugs right now target a specific protein, HER2, EGFR, all these different nomenclatures. The bottom one, PLX, that's targeting that mutant BRAF. And by doing so, it's blocking this protein in only patients that have this mutation. So it's going in and blocking a mutation that's only present in cancer cells, which is distinct from the normal cells. So if you can go in and specifically just target the tumor and not any other cells in the body, that's a huge goal right there. So right now, this medicine, showing you in the bottom right-hand corner just a picture of stuff that I look at every day, people's genetics on a computer screen, displayed as a figure right there. It was in the New York Times 2 months ago. This is late-breaking research that this medicine is doing so well in patients that it'll likely be approved pretty soon. So last two slides, I told you a lot about cancer biology, and I really want you to understand where I'm coming from. The reason why people go into drug development in general, it's a long process, it's very hard. When you study biology and math in school, it's not easy. But for me, I mentioned before that I'm very close with my family. So I never met my mother's mother. She passed away from cancer when before I was born. My father's mother, my grandmother, also passed away from cancer when I was about 12 years old. So right off the bat, I kind of combined my scientific interest with my passion for my family, and was like, okay, well, if I could use that to get into disease treatment somehow, in some way, maybe I can contribute to a company like this and make a difference. So for me, it's very personal. And going to work every day, I think about this all the time. Mark Fanian is a colleague of mine. So I told you about all the volunteer work I do, and going out and speaking to students and how much it means to me. Mark's a great guy. He helped me organize these events. He's not a scientist. He worked in corporate communications. And he would help me organize everything and make sure that whenever I went to an event like this, everything was in place. Mark, as you can see, beautiful wife, beautiful children, and it's not a positive story. We lost Mark earlier this year. He's a friend of mine, and was a colleague of mine, and he passed away from cancer this year. So with every single time I come out and speak about cancer research, I remember him and remember that he was only a few years older than me. So it's very, very personal for my family, for my work family, and maybe for some of you as well. Are there any football fans out there? NFL, professional football? Me too. I love it. Love it. My team isn't doing so hot this year, but I can talk to you guys about that later. I want to tell you one last thing, and that's about a man named Elijah Alexander. So for all of you who don't know what a linebacker is one of the toughest guys out on the football field. He's on defense. He's the one who goes and tackles everyone with the ball. Elijah Alexander he retired from the league about 10 years ago, and he was a linebacker for the Raiders. He played for the Colts. When he retired from the league, he came down with multiple myeloma, type of blood cancer. And he actually went on the drug that my company makes, Velcade, to try to treat that cancer. Now, about 6 months ago, Elijah came to my company during a company meeting, 1,200 people out in the audience, to speak to all of us about the work that goes on at a cancer research company. And this guy, he's a linebacker. He's a massive guy, big, strong, passionate. And when he got up and spoke at this event, big booming voice, he commanded the entire room. And with the love and affection that he spoke about his family, I'll never forget it. I will never forget it for as long as I live. So he spoke about his family and about what it meant to come down with cancer when he retired from the league. As you can see, he's got a beautiful wife who I had the privilege of meeting just about 2 weeks ago at an event. Again, the reason why I mention all this to you is because this didn't have a happy ending either. He came to speak with us 6 months ago. We lost Elijah and Mark within about 2 months after that. And this is from a disease that we're looking to treat. We're able to extend his life, but the work isn't done. It's not even close to being done. And that's why coming out and speaking with all of you is so important to me, because it's the next generation of scientists and researchers out there who are going to have the biggest difference. As you can see with the targeted therapies, we're getting closer. Every single day we get closer to understanding the different genetic mechanisms behind cancer and understanding how we can treat patients better. But we're not there yet, and I'm hoping that a lot of you, through your scientific studies, will come and help us out. So with that, I want to thank you again for having me, and I'm pleased to take any questions. Thanks a lot. And I know we're running short on time, but please, yes.
I was wondering, how breast cancer develop? Why do different mechanisms to breast cancer? Nobody in my family has it, but my aunt recently just got diagnosed with breast cancer. That's out of the random. How does that happen?
So it's a great question. For all of you that didn't hear about it, the young lady over here doesn't have a history of breast cancer in her family, but a family member was recently just diagnosed with it. So as we mentioned before, it can either be a family history or it can be environmental factors that play a role in it. It might not even be that too. What causes cancer is DNA damage and random mutations. So it could be a random set of, and it's not just one mutation that's going to set off this cascade of cancer. It's mutations build up over your life. That's why you'll find that cancer usually hits an older population, but people our age get it too. One of my good friends passed at 23 from it. So random mutations, and it could have been some of those environmental factors that we mentioned before, but it's impossible to speculate without the doctors probably know and are studying that. Let me get some of the other ones. Yes, you right here, please. Yes.
Are you more likely to receive the cancer from your mom's side or your dad's side? Because my dad's side, every single person on my dad's side got cancer. So
As far as I know, it's random. I don't know if there's a bias between men and women. I just made that joke earlier about women, but as far as I know, it can either your mom's side or your dad's side can be affected by it. Personally, I've noticed it on both sides for me. So it is random, and if you notice it on one side of the family, well then they have your dad's or mom's side has the history, whereas the other side may not, and it influences your personal risks.
okay, 2 questions. First one, how long has your company been working with PLX?
So that's a good question. We don't. That's not a Millennium molecule. That's a company called Lexicon. And they were just recently acquired by a bigger company, and I believe it's Merck, but I don't remember. Or it might be Roche. If you Google Plexicon or PLX, that'll definitely be one of the first things to pop up. Second question.
The second one, what's your football team?
I am a loyal. So as I mentioned, I grew up around New York City. So all of my friends were Jets and Giants fans. Now, the way when I got into football, I was between 8 and 10 years old. I was like, well, wait, the Jets and Giants, they play in New Jersey. I'm a New Yorker. How am I going to root for a team that plays in New Jersey? So I started rooting for the Buffalo Bills, who play within the state confines of New York. It's 500 miles away from where I grew up, but loyal fan, you got to stick with the team. Tough year.
Is there a way for you to test for the BRCA genes?
Yes. So these are distinct mutations within these genes. And I haven't looked at these ones specifically, but being in the clinical department at work, where we work with patient samples all the time, we test for mutations. So in 2003, the human genome sequence was uncovered. So we basically understand the genetic code within all of our cells and all of our bodies. And we can go in and test for different mutations like that. Yes, please.
Is there a way to speed up phase 1, phase 2? The idea being that phase 1 patients are at a point where immune compromised and extremely ill. Is that really an accurate way to see what the genetic basis is to do biopsies for phase 2?
So there are ways, yes, to accelerate the phase 1 trials, and I think companies think about it all the time, because the ultimate goal is obviously to get the patients to the correct patient population. So certain designs behind the phase 1 trials, as far as reaching your MTD, maximal tolerated dose, quicker, so we can put it into the appropriate patient populations in the phase 2s. Even some of the phase 1s now have specific stages, sort of like a phase 1/2, that where you can start getting it into patients a little sooner. Yes, please. Your likelihood of getting cancer. So, we spoke about a few of them with, cigarette smoke. So you control what goes into your body. That definitely increases your risk more than any environmental factor that I've ever seen. UV sunlight, so going to the beach. I used to be a lifeguard and swim instructor, so you have to put on the sunblock. Different things like that we had in the slides earlier. One more if anyone's got one in the corner. So that's a very good question. And again, I spoke about a lot of my friends in high school who smoked. The only good thing about smoking is that it's reversible. So if smokers who are out there, they've been doing damage to their lungs for who knows how long or how many years. Stopping, it starts to clear up. So when you go into doctors for a doctor's exam, you can see they do some imaging. You can see what was previously maybe a black lung starts to have that pinkish shade to it again. So it's not a definite that someone would develop cancer. In fact, being at such a young age, better to stop sooner than later. Good question though, because it absolutely is reversible. Maybe those mutations haven't occurred yet. Thank you very much. I appreciate it. Good questions.